Before your child speaks their first word, before they take their first step, before any teacher, doctor, or parent shapes who they become, something else is already at work. Trillions of living organisms, invisible to the naked eye, are producing the exact molecules your child's brain needs to wire itself correctly. They live in the gut, they are bacteria, and the window in which they do their most important work closes before your child turns 3. What happens inside that window, what feeds these organisms, what kills them, what replaces them, is one of the most consequential biological events of your child's entire life. This is not a theory. This is peer-reviewed science from the Lancet, Nature Communications, and the National Institutes of Health, and most parents have never heard a word of it. Let's start with what scientists now call the gut-brain axis. For most of medical history, the gut and the brain were treated as separate departments. The gut digests food, the brain thinks. That was the understanding. It was wrong. Research published in Frontiers in Nutrition in 2025 confirmed that the gut and brain are locked in constant, bidirectional communication, and that this conversation is especially critical during the earliest years of life. The gut sends signals upward through the vagus nerve, through the immune system, and through chemical messengers in the bloodstream. The brain responds. Both systems develop together. Neither is fully independent of the other. Now here is the part that changes how you think about early childhood. A child's gut microbiome begins forming before birth and reaches its foundational structure by age 2 to 3. During that same window, the brain grows to roughly 80 to 90% of its adult volume. These timelines are not a coincidence. They are synchronized, and emerging research suggests that synchronization is deliberate. The mechanism works like this. Bacteria in the gut ferment dietary fiber and produce compounds called short-chain fatty acids, butyrate, acetate, and propionate. These molecules enter the bloodstream, cross into the brain, and trigger the production of a protein called BDNF, brain-derived neurotrophic factor. BDNF is the brain's primary growth signal. It tells neurons to develop, branch, and form connections. It drives the process of myelination, the formation of the protective sheath around nerve fibers that determines how fast signals travel. It supports neurogenesis, the creation of new brain cells, and the consolidation of long-term memory. Animal studies show this pathway clearly. Emerging human data supports the same direction. When BDNF production is compromised in early life, the architecture of the developing brain is affected, not cosmetically but structurally. 2 bacterial families appear most critical in this process. Bifidobacterium and Bacteroides, found consistently in healthy infant guts, show the strongest association with cognitive development, motor milestones, and neurotypical brain structure in early childhood. These are not background players. They are central to the construction project. Now consider what is happening to the microbiomes of children born in America today. Infants born vaginally are colonized during delivery by their mother's lactobacillus, Bacteroides, and Bifidobacterium, founding populations that seed a healthy ecosystem. Infants born by cesarean section miss that exposure entirely. Research published in scientific reports found that Bifidobacterium and Bacteroides levels in cesarean-born newborns were significantly lower than in vaginally delivered infants. More than 32% of American births are now cesarean deliveries. To be clear, cesarean section is a life-saving procedure. This is not an argument against it. The point is narrower and more specific. The microbiome difference is real, it is documented, and it statistically increases certain developmental risks. It is not a sentence. It is a variable that deserves attention. The disruption extends well beyond the delivery room. The CDC reports that the United States prescribes antibiotics to young children at rates among the highest in the developed world. A significant portion of these prescriptions are written for viral infections, common colds, ear fluid, mild respiratory symptoms, where antibiotics have no clinical effect on the illness itself. What they do affect reliably is the microbiome. Research from Translational Pediatrics demonstrated that early life antibiotic exposure reduced myelin basic protein expression in developing brains, a measurable structural effect, not a theoretical one. Disrupted myelination affects processing speed, learning, and coordination. These are not abstract risks, they are observable outcomes. A 2025 study published in Nature Communications found something equally striking. The composition of a child's gut microbiome at age 2 was associated with their emotional regulation and brain network connectivity years later, in middle childhood. The bacteria present at 2 years old were predicting behavior at 10. Here is the part of this story that medicine has been slowest to absorb. When a child is diagnosed with ADHD, anxiety, or developmental delay, the standard workup looks at the brain. It rarely looks at the gut. The microbiome is almost never evaluated. The dietary factors that determine what bacteria thrive or starve are almost never discussed during a pediatric appointment. Yet the research shows that gut dysbiosis, an imbalance in microbial communities, is consistently associated with autism spectrum disorder, ADHD, Tourette syndrome, anxiety, and depression in children. The association is not speculative. The converging evidence from multiple independent research groups is strong enough that leading neuroscientists now treat it as a serious causal hypothesis, pending the completion of long-term human trials. What this means practically: the gut is not a bystander in childhood neurological development. It is an active participant, and in millions of American children, it is operating in a compromised state, shaped by delivery mode, formula feeding, early antibiotic exposure, and diets built around ultra-processed foods that provide almost nothing for a microbiome to work with. The brain does not develop in isolation. It develops downstream from the gut. That changes the conversation entirely. So what does the evidence actually support? First, breastfeeding, where medically and personally possible, remains the most evidence-backed early intervention for microbiome health. Breast milk contains specific fibers that selectively feed Bifidobacterium infantis, the dominant strain in a healthy infant gut. For mothers who cannot breastfeed, newer probiotic supplemented formulas show genuine promise in recent clinical data. The goal is the same regardless of path, protecting microbial diversity in the earliest months. Second, dietary fiber throughout childhood is the raw material for SCFA production. Whole fruits, vegetables, legumes, and intact grains feed the bacteria that produce butyrate and propionate. Ultra-processed foods eliminate this input almost entirely. This is not a minor nutritional preference. It is a neurological consideration. Third, unnecessary antibiotics carry a cost that pediatricians rarely quantify in the moment of prescribing. If your child has a viral illness and is not in distress, a direct conversation with your doctor about watchful waiting is both reasonable and supported by current evidence. You are asking the right question. Finally, if your child shows persistent gastrointestinal symptoms alongside developmental delays, behavioral regression, or signs of chronic inflammation, ask about a referral to a pediatric gastroenterologist. The gut-brain connection in these cases is not a fringe consideration. It is increasingly central to how progressive pediatric medicine is approaching neurodevelopmental conditions. Your child's brain is not built by genetics alone. It is built by what happens in their gut in their first 1,000 days before most parents know to pay attention. Now you know.